Okay, in this video I'm going to show you how to do some simple chromosomal mutations, what they look like on paper. So you should have either this slide of your notes open or turn off this worksheet and do it from this worksheet. Either way is fine. So before we begin, make sure that you have your chromosomes labeled A, B, C, D, E, F, G, H all the way across for all four of these chromosomes here on the left. So go ahead and pause the video, write those down. And then for each one of these, what you're looking at is the difference between the normal chromosome, what should be happening, and that's on the left side, and on the right side is the mutated chromosome. So let's start with the first type of mutation. That is called deletion. And the name of the mutation explains itself. So instead of having our letters A through H, we're just going to take a chunk out, and we'll say that chunk will be letter C. So now our chromosome reads A, B, D, E, F, G, H, where letter C is missing. And these chunks of chromosome I'm referring to could be any number of base pairs, thousands, tens of thousands of base pairs. Okay, so letter C is missing right there. So basically what happens is that chunk of chromosome C gets removed and then just kind of disappears. The next type of chromosomal mutation is called insertion, which is basically the opposite of deletion. So we're going to take, again, just focusing on the same area of chromosome. It could be any part of these chromosomes that this is happening to, and most likely the end of them. But we're going to take B and C and basically replicate it. So a B and C chunk of chromosome got inserted right there. So we have two chunks of B and C. The rest is the same. So in this case, information gets added. In the previous case, there's information that is actually missing. Both are bad or harmful. The third type, you're going to see, again, I'm just going to pick the same kind of chunk of chromosome. This could be any chunk. Let's pick B, C, and D. Third type is called inversion. So what happens, we still have the same amount of information, unlike the other two, but now our information is flipped around. Which still can be harmful, because if the information is supposed to be read in order, make certain proteins, and they're read out of order, it could be a big deal. The last type of chromosomal mutation involves actually two chromosomes. So let's pretend for this first three example we've been talking about chromosome number 1. And for this example we're going to use two chromosomes, so let's pretend we're talking about chromosome number 1, but also chromosome number 19. Which has different information. So a completely different set of genes found on chromosome 1 versus found on chromosome number 19. This fourth type of mutation is called translocation. And again, as the name implies, it looks like we are moving something to a different spot. So let's just take this part right here, WX, is going to come out, but instead of going missing like it did in deletion, this part of chromosome is going to get put into a different chromosome. So now our chromosome reads A, B, C, D, E, which is normal. We've added WX, coming from chromosome number 19. So here are your 4 different chromosomal mutations. They pretty much explain themselves, but as long as you can understand how these work in a picture, show that these are got flip-flopped. Oops. These are our 4 different chromosomal mutations. Deletion, where one is missing. Insertion, where information gets added. Inversion, where information gets switched around. And translocation, where information from one chromosome winds up on a different chromosome.